Dear students, welcome to the massive open online course on plant diversity. Today we are going to discuss on some basic aspects of viruses. Now first of all, we should know what is a virus. Virus is a small infectious agent, also known as obligatory parasite, as it can replicate only within the host cell and it has a genome of either DNA or RNA. Now we should know how the study of viruses, that is virology, became so relevant. It was observed that many of the viral diseases of plants and animals lead to huge economic losses unless effectively controlled. Secondly, some cancers are of viral origin. Very few effective and acceptable drugs of viral diseases are present till today. Some of the viral vectors are now used in gene therapy. Critical examination on viral host interactions led to fascinating insights into genetic mechanisms. And lastly, viral-based vaccines are a promising approach as targets against various infectious and malignant viral diseases like hepatitis C and HIV. Now, towards the last century, the pathologists became very confident about the germ theory of disease, that each and every microorganism is associated with a specific disease. They can be seen with the help of a microscope, they can be cultivated on a nutrient medium, and they can be retained by filters. But these observations were opposed by some conflicting observations. Now what were those? In the year 1892, Dmitry Ivanovsky found that a bacteria-free filtrate caused the mosaic disease of tobacco plant. A bit unimpressed by his discovery, but that was being confirmed by Bejering in the year 1898, who termed that bacteria-free filtrate as Contagium vivum fluidum, later termed as virus. In the same year, another important discovery was made by Loeffler and Frosch. In case of foot and mouth disease, it was being transmitted from animal to animal with great dilution at each passage. So it had to be a reproducing agent and could not be a bacterial toxin. Soon many of the discoveries followed. In the year 1908, Ellerman and Bang reported cell-free transmission of chicken leukemia. In the year 1911, even another important discovery was made by Rouse. He found solid tumors on chicken and it was transmitted by cell-free filtrate. Now this was the first indication of viruses causing cancer. Now in the year 1915, Todt was trying to cultivate smallpox agent on a nutrient medium and suddenly there was a micrococcal contamination. Now those micrococcal colonies took glassy appearance and no bacteria could be subcultured from those glassy colonies. Now if those glassy material was added to normal colonies, those also took the glassy appearance. Later on, these were termed as bacteriophages. Now similar observations were made by D. Harrell in the year 1917 in case of dysentery bacilli. He took a bacilli lawn and filtrate of fecal emulsion when he obtained clear spots. So these were finally termed as bacterial viruses or bacteriophages, and from these observations finally initial definition of virus came out, that it is a small infectious agent that could not be seen by an ordinary microscope. It cannot be cultivated in absence of cells and cannot be written by bacteria-proof filters. The question that comes next, that whether viruses are living or non-living. Now from living organism we can say that it is an ensemble of organized biomolecules in a specific configuration that has the ability to grow and proliferate. So by this criterion, viruses do not qualify as a living entity, as it can grow within the confinement of a cell which is itself an independent entity. Parasitism also does not qualify a virus as a non-living entity. So finally, Luria and Darnell define virus as an entity with genome of either DNA or RNA and which uses host cell machinery to make virus-like particles to form progeny variants, and their genome can be transferred to other host cells. For example, TMV in the purified and crystalline form can behave as a non-living entity, and when it affects a tobacco plant, it behaves as a living entity. So viruses are finally termed as an intermediate between living and non-living. So having discussed the discovery of viruses, now let us go straight into the structure of viruses. Viral genomes are actually surrounded by protein subunits called capsid. The unit of capsid is capsomere, and even further subunits are called protomers. So what are the functions of these protein subunits? These protein subunits actually protect the genome from nucleus degradation and shearing. They also contain ligands that can bind to receptors on host cell surface in order to infect. Exception being the plant viruses, which can cause infection upon direct injury. These proteins also contain enzymes required for infectivity. They also contain a genome release system that releases the genome at proper target cells. Now the interesting fact is that all these protein subunits, as we know that the secondary structure of protein is a regular structure like of alpha helix and beta sheet, but when it forms a tertiary structure, it becomes asymmetrical. Now individually these asymmetrical subunits form a symmetrical viral particle so as to get maximum subunit-subunit interaction and a minimized free energy state, and hence it forms a regular stable structure. So there are various arrangements of these asymmetrical protein subunits. Now let us have a look into this filamentous type of virus, where we can see that individual protein subunits, which is asymmetric in nature, together form a symmetrical stacked disk 2D structure. Now if this stacked disk 2D structure is placed on top of one another, they will form a hollow tube with a room for the nuclear genome. For example, a closer look at the virus tobacco mosaic virus gives a helical capsid which fits a helical nucleic acid within it. So examination of all the filamentous viruses so far have seen that all are helical. Now there are viruses with icosahedral structures. So what is an icosahedron symmetry? An icosahedron is made from 20 equilateral triangles. That is, we can see that 5 at the top, 5 at the bottom and 10 around the middle. It has 30 edges and 12 vertices and requires 60 protein subunits at the 12 vertices, and hence they form a group of 5 protein subunits at each of the vertices. It has 3 axes of symmetry, 5-fold, 3-fold and 2-fold axis of symmetry. Now this slide depicts that at each of the vertices of the original icosahedron, there are a group of 5 protein subunits forming a structure called pentamer or penton, and a ring of 6 subunits at the new vertices, which is called hexamer or hexon. Now there are still other viruses which are enveloped. Now where from does this envelope come? These envelopes are derived from host cell membranes during viral budding. Now various forms of envelopes are there, like this one, which is an isometric core with an isometric envelope, for example Synbis virus. A helical core with a spherical envelope like that of influenza viral particle, and a helical core with a non-spherical envelope like that of rabies virus. So, still there are other diverse forms of virus like that of head-tail morphology, which is also known as binal symmetry. What is that? It is nothing but a head with a icosahedron symmetry and a tail with a helical symmetry. Now, there are other structures also like tail fibers, base plate, callus, and these type of viral particles are classified on the basis of tail length, contractile or non-contractile tails, and other features. These viral particles actually attaches to the host cell surface through these tail fibers and injects the genome, which is tightly packaged within the phage head with using tail as a conduit. Now we can clearly see how complex are the structure of viruses, and we can clearly imagine also how difficult it would be to classify these diverse form of viruses. So classification is a very complex task, and any new classification of virus can predict its replication, pathogenesis, and transmission. Now 5,000 viruses have already been catalogued and many more are in the pipeline. Viruses are diverse with respect to the diseases they cause, their host range, morphology, and nature of genome. Now if we come to the first feature, that is the diseases they cause, we can see that viruses are anthropomorphic, that is those viruses which can infect only humans and livestock. One virus can cause the same disease in different hosts, different diseases in different hosts, or no disease in certain hosts. Viruses with different molecular makeups can cause the same disease. So it's very difficult to get a representative virus, as we can see. Now classification on the basis of host range, we can say that viruses have unique hosts, narrow range of hosts, or broad range of hosts across plant and animal species. The same host can be infected by several viruses with little similarity in molecular makeups. In terms of particle morphology, we can say that viruses, even two viruses with similar morphology, can have totally different biology, pathology, and molecular biology. Lastly, with respect to genome, we can say from Hershey and Chase experiment that the nuclear genome contains all the genetic information necessary to form progeny variants. So how can we classify virus with respect to the nature of genome? That can be classified on the basis of the presence of DNA or RNA, and whether those DNA or RNA are single-stranded or double-stranded. Now whether DNA or RNA, that can be detected by determining the base composition, the buoyant density, and sensitivity to DNAs or RNAs. And whether single-stranded or double-stranded can be detected by hyperchromic shift, and whether they follow Chargaff's rule. The key aspect of all this classification is mainly the nature of genome, the replication strategy and pathogenesis. Now there are two main mode of classification of viruses. One is Baltimore classification, another is ICTV classification. So let us have a look on the Baltimore classification. Now the Nobel laureate David Baltimore have classified the virus putting stress on the mRNA and their encoded viral proteins and the replication strategy. So he has divided these viruses into 7 major classes based on the type of DNA present and the replication strategy. Although there are some demerits of this classification, like two very different distant viruses like animal virus variola and bacteriophage T2 both contain double-stranded DNA, but they are very different in their molecular makeups. So that is not being considered in this classification. And also in the class 4 and class 6 virus, there are some ambiguities. So let us have a look on a detail in this type of classification. So this slide actually depicts the 7 classes of virus. So let's have a look. Class 1 virus comprises of double-stranded DNA and includes adenoviruses, herpes viruses, pox viruses. Class 2 virus comprises of single-stranded DNA and it includes parvoviruses. Class 3 includes double-stranded RNA and examples are reoviruses. Class 4 comprises of single-stranded RNA that is of positive sense. Now we should know here what is a positive sense mRNA and what is a negative sense mRNA. Now positive sense mRNA is that mRNA that can be directly translated into proteins, and negative sense mRNA is that mRNA which is complementary to the positive sense mRNA and can never be directly translated into proteins. So this is the basic difference. So class 4 virus comprises of positive sense single-stranded RNA. And the examples are picornaviruses and togaviruses. Class 5 comprises of single-stranded RNA, as I have told. This is of negative sense RNA. And the examples include orthomyxoviruses and rhabdoviruses. Class 6 viruses comprises of single-stranded RNA, and the speciality here is the presence of reverse transcriptase. So it gets converted to mRNA through a double-stranded DNA intermediate, and this form is being made with the help of the enzyme reverse transcriptase. Now the last class, that is the class 7, it comprises of double-stranded DNA. So how it differs from class 1 virus? That also comprises of double-stranded DNA. Now here there is a unique feature, that is this double-stranded DNA is replicated through a single-stranded RNA intermediate and includes hepadnaviruses. So this explains in totality the 7 classes of viruses as explained by Baltimore. Now there is another mode of classification, that is ICTV, which has taken into account these features like morphology, nature of genome, host range and other special features so that they can put a virus into specific taxonomic group. So this slide depicts the hierarchical classification including all the phylum, subphylum, order, class, family, etc. up to species level. So we can see here, for example, a family ends with suffix viridi and a genus ends with suffix virus. So we can tell that in the Retroviridae family there is lentivirus genus with species like HIV1, HIV2, SIV, BIV, FIV, and HIV1 has different types of strains depending on the target cells they attack. So as of November 2018, there is a single phylum 2 subphyla, 6 classes, 14 orders, 143 families, 846 genera, and 4958 species of viruses as explained by ICTV, that is International Committee on Taxonomy of Viruses. So finally, this is a detailed summary of the classification of DNA virus with respect to various properties like symmetry of capsid, genome architecture, variant polymerase, variant diameter, genome size, and similarly, this is a detailed classification of RNA viruses. So, to summarize, today we have discussed about the discovery of viruses, their structure and classification. So, in my next lecture, I will continue with this. I will explain the different replication strategies of the different classes of virus. That's all for today. Thank you.